I'm delighted to be able to speak with y'all today. As Ellen mentioned, I am an alum of the department, so it's nice to see many familiar faces and names. I'm just excited to share with you some of what I've been up to recently. So I'll be presenting today on some of the research that I did together with some colleagues in forestry and natural resources, as well as several students in forestry, wildlife, and NRES over the summer. And so this is kind of a preliminary results presentation. That's your heads up. Stay tuned for more detailed results over the next year or so. So let's jump in. So I'll be talking today about Reforest the Bluegrass sites. For those of you who may not be familiar, Reforest the Bluegrass is a community urban reforestation program that is run by LFUCG Environmental Services, and I think we have some colleagues from there who are with us today, and thanks so much for joining. So Reforest the Bluegrass was kicked off in 1999. The first planting was done then, and they have planted a site every year. They are really good at involving community members and participating in urban reforestation. So they've had over 17,500 volunteers. Oops, it just advanced on me. Over 17,500 volunteers over the duration of the program. And they've planted over 150,000 trees on 20 sites for an estimated 195 acres of urban reforestation. And these sites are also actively managed by LFUCG Environmental Services. So they are continuing to do work in these sites to help them to continue to develop on a desirable trajectory. And we'll talk more about that in a little bit. So what you need to know is there are 20 Reforest the Bluegrass sites across Lexington, and they were planted at different, one planted every year. So we're essentially working with a 20-year chrono sequence of planted urban forests, which is a really interesting and cool research opportunity. So in this project, I was interested in a few key questions. And I'll be basically talking through each of these step by step. So first, interested in the plant community and how that is developing over this chrono sequence, and including both woody plants, overstory and understory plants, as well as the herb layer in that. Interested in soil development over time in this chrono sequence, as well as the possible effects of riparian reforestation for the sites that have streams flowing through them, the effects of that riparian reforestation on water quality, seeing if there's possibly a water quality improvement as streams flow through the sites, and then also whether birds are using these reforested sites and to what extent. So getting some data related to bird communities on these sites. So a quick overview. I've already mentioned Reforest the Bluegrass sites are scattered across Lexington, and on this slide, you can see the red patches on the map of Lexington indicate where Reforest the Bluegrass sites are located. And then to the left is a map of the Reforest site at the Kentucky Horse Park. And the pink dots in that map indicate where our plots were established. So I'll go ahead and introduce that protocol. So we received approximate site boundaries from LFECG at the beginning of the project, and then some of my awesome interns, Kiernan and Whit, went through and digitized those from the kind of approximate boundaries to what seemed like more delineated actual project planted boundaries on GIS, and they developed a shapefile from those data, which was really great. We used those data to, and on this horse park map on the left, you can see there's the red boundary around the outside. That's the planted boundary, and then there's an additional green line that runs in on around the inside of that. That was our buffer. We established a 65-foot buffer from the site edge to try to minimize edge effects. And then we set up permanent monitoring plots within that buffered area. So these pink dots in the horse park site represent where our monitoring plots were established. Each monitoring plot, we set up a 50th acre plot and a 200th acre plot. In the 50th acre plot, we took overstory measurements, and in the 200th acre plot, we took understory measurements, and those were concentric on the plot center. Okay, so to jump into specific methods, first, we will talk about trees and shrubs, the woody plants. And here we've got photos of Kiernan and Whit, who I mentioned earlier, a couple of the interns who worked with me over the summer. So our overstory data, we surveyed trees that were greater than 1 inch in diameter in the 50th acre overstory plots. And for that, we recorded species, height, and DBH. And we also surveyed trees and shrubs less than 1 inch in diameter in the 200th acre understory plots. And we just recorded height and species for those. And these are some of the key results from that. And thanks to Zach Hackworth, who collaborated with us on this, and John Lotka, these figures are part of a manuscript that we recently submitted to a special issue of the Journal of Forests. And so fingers crossed for favorable reviews on that. A couple key things that I wanted to talk about in these figures. The figure on the left, the blue bars represent the height of native species in the understory, and the red bars indicate the height of invasive species in the understory. And so the key thing that I wanted to point out on this figure is just that over time, the understory, at least in the older sites, the understory is increasingly dominated by invasive species, and the native species are being topped out there. And then in the younger plots, the native species, for the most part, seem to be higher than the invasive species in the understory. So an interesting pattern there to continue to follow up on. Definitely invasive species are present and problematic in all of these sites. And so that represents a really significant concern for management. And luckily, LFUCG is doing ongoing management for invasive species on these sites. So over time, hopefully we'll be able to bring that invasive species presence down in the understory and have a better and more robust native species presence there. On the right, bottom right of this slide, you'll see upper canopy height regressed with planted year. And so what I want to point out about this figure is just this really interesting logistic relationship of the upper canopy height with planted year or time since planting. And so we, John Lotka and Zach and I were scratching our heads trying to figure out what was going on with this, this figure, these data. And we settled on a couple interesting possibilities. So one possible explanation for these data, and I should mention real quick, we specifically what I'm talking about here is just this flattened height growth in the older sites. So basically from 2005 to 2000, the height is fairly consistent. So it's kind of curious that the canopy height isn't continuing to increase in these older sites. And that's the curious thing that we were scratching our heads about. So we settled on a couple possible variables that might explain that. One thing is that the canopy in some of the older sites was dominated, at least in the beginning, by ash species and black locust. And we're noting in some of the older sites, as would be expected, that the ash trees are uh, not being successful anymore, that they're declining and dying out. Um, and so that could be part of what we're seeing in some of these older sites, that the trees that were on the leading edge of that canopy growth for the first several years after planting, um, those are dying out. In the case of the ash, because of ash borer. Black locust, the cause is less well known, but those are disease prone and pest prone trees. So it's not surprising that they are also um, declining at this point. And then the other possibility is that some of the trees that are present in the canopy in these sites and some of these older sites are actually not not typical canopy forming species in climax forests in this area. So we noted species like box elder, which are present in the canopy in some of these older sites. And in more mature forests, those will be um, less of an influence in the canopy. So we're expecting that over time, this curve may start to increase again. We might start to see some increased canopy growth in some of these older sites as as the trees that are currently present in the canopy that are dying off as they continue to decline and be replaced by trees that will continue to form the canopy. So, for example, in a number of the sites, these older sites, we did see some good oak advanced regeneration. And so we're anticipating that those oaks will come in and start advancing that canopy over time. Herbaceous vegetation. We sampled the herb layer in 10 2-foot by 2-foot grids, randomly placed in the overstory plot, and then recorded percent ground cover by each species that was present, as well as bare soil and litter as appropriate. The preliminary results from herbaceous vegetation data, and thanks to my intern Julia, who pulled these data together, and who will be helping to do further analysis on these data and hopefully work up a manuscript soon. So we found that species richness in the herb layer ranged from 16 to 43 species across the sites. Invasive species were present in the herb layer at all sites, and some of the most common included garlic mustard, ground ivy, and winter creeper. And then in some of the sites, the importance of or the presence of some of these invasive species were very significant. So for example, more than 50% of the ground cover at one of the sites was comprised of winter creeper. So definitely a really significant invasive species problem in the herb layer in some of these sites. In addition to the aforementioned invasive species presence in the shrub layer. So that'll be an interesting pattern to continue to track over time, especially with ongoing management activities. We also noted, as would be expected, a shift in species that were present from shade intolerant species to more shade tolerant species as we move through the chronosequence from the new sites to the older sites. So for example, 25% of the taxa observed at the 2019 site were grasses and grass allies, but only 4% of the species observed in the 2001 site were grasses and allies. So we're seeing shifts in species composition as would be expected as the sites develop and shade is more of a factor. So much more work to be done with herbaceous data. I wanted to give a quick preliminary overview of those. Preliminary, so for onto birds, bird communities were surveyed using 10-minute point count surveys in early June of 2020. And species richness and diversity were calculated from those data. And this is a photo of Kaylee. Kaylee and Sabrina were my awesome interns who specialized in birds, and they did really great work. And these are some of our preliminary results from the bird surveys. And I should note, and I'll mention this again at the end, but we do have some additional proposals out to get some funding to do some more detailed and comprehensive bird surveys to get a little bit more of a robust sampling of the bird communities across these sites. So stay tuned. Hopefully we'll get some of that funding and be able to build out these analyses a little bit more into the future. But some of our preliminary data, I wanted to show that the bird community, bird diversity seems to have a quadratic relationship, a weak quadratic relationship with stem density in both understory and overstory spaces, as well as overall. And again, these are certainly weak relationships, and we'll need to do some additional sampling to get a more detailed analysis of some of these relationships. But these relationships will be consistent with the literature that suggests that that stands that have too high of a stem density are unfavorable to birds, especially particular bird species. So we'll have to do some additional surveys and follow-up analyses to more clearly define this relationship. I also wanted to just point out this very weak linear relationship between the planted year and bird diversity. So very weak, non-significant, I'm sure, but a very slight increasing trend from the newest sites, the newest site planted in 2019 to the oldest sites planted in 1999 and 2000, which was part of something that we expected to see. We did expect to see an increase in bird community bird diversity as we moved through the chronosequence with higher diversity in older sites, especially as those sites develop critical kinds of habitat and provisioning for a variety of bird species. So we'll have to continue to monitor that over time, and I think those follow-up surveys will be really helpful to characterize some of those patterns more clearly. For water quality, we did a very simple water quality analysis. We collected grab samples from streams upstream and downstream of the sections that were planted as part of Reforest the Bluegrass. And then Millie in the Forestry lab analyzed those for a series of water chemical characteristics. I'm only going to talk about nitrate today, but these are also preliminary results, and we're doing some additional work on these and have another proposal out that will hopefully get funded to support some ongoing water quality monitoring on these sites. But these are figures that show data from the 4 sites that we picked for monitoring. Each of these sites has a stream that flows through it and was planted as part of Reforest the Bluegrass. I'll note quickly that the Horse Park site, that stream was not permanent. It did dry up during the summer, and so we had a much smaller sample size from the Horse Park site than the others. But I wanted to just point out in the Hickman tributary, as well as the main stem of West Hickman, we did see a significant difference in nitrate concentrations between the upstream sampling site and the downstream sampling site. And this is an interesting result, then I think we'll have to do some follow-up work to further characterize that. But that suggests that the nitrate levels are being attenuated slightly as the stream is passing through the reforested reach. So that would be a really great result if that's something that bears out with in our more robust sampling efforts, because that would suggest that this kind of small scale riparian reforestation can have some effect on in-stream nitrate concentrations, which should be really great in thinking about reducing nitrate exports to, for example, the Gulf of Mexico. Moving on to soil chemistry, we collected soil samples from each plot in each site. So there were at least 3 plots in each site and up to 9 plots in some of the sites that were larger. So we collected a composite sample from each of the plots within each site and analyzed those for a suite of different characteristics. And I wanted to show just the ones that demonstrated a significant relationship with time since planting. And so I'll orient you to these figures. They are set up a little differently than some of the other ones. So on the x-axis is years since planting, and on the y-axis is the different variables that we measured. So on the top left is silt, and we noticed that percent silt is increasing across the chronosequence. So higher percent silt in the oldest sites, and lower percent silt in the higher sites, or sorry, in the newer sites. Phosphorus also shows an increasing relationship with time. So highest phosphate levels in the older sites, as well as zinc. So increasing zinc in the older sites. And then I included here carbon. Carbon did not show a significant relationship with time, but I included that figure here because I thought that was interesting that carbon didn't show a significant relationship with time. I definitely anticipated that percent carbon would be increasing across the chronosequence, that it would be accumulating in these soils. And the lack of that relationship, I think, merits some additional work as well. And maybe there may be some site history and topographic and other sorts of variables that explain that variability. So our current work that's in progress, I mentioned the trees and shrubs manuscript that is in review, as well as the herbaceous vegetation data that Julia and I will be working with this semester. We also submitted a proposal to the Kentucky Water Resources Research Institute and hope for favorable results there, and that will support continued water quality sampling, including some macroinvertebrate community sampling, as well as engagement with some local schools. So we're excited about that possibility. We've submitted a small grant, the Monroe Award, and a NIFWIF proposal for doing some additional bird habitat improvement and bird community assessment work, as well as doing some additional community engagement on engaging the community in reforest sites. And those are both in review currently, and we're hoping for good results there. Also working with Dr. Crocker on developing a survey and kind of a video tour to engage the community in better understanding their community perspectives on reforest sites and community interest in participating in like service on reforest sites. So we'll hopefully have that ready to roll soon. And then I've included here at the end, I think some additional work that really needs to be done on these sites includes some additional surveys to characterize other taxa that we haven't yet surveyed. So I think insects and spiders would be really great to look at across the chronosequence, as well as herps, especially in the sites with streams. Those data would be really helpful to collect and to better understand how these sites are developing over time. I'll real quick mention my the many people that I have to thank. I've mentioned some of them already throughout the presentation, but funding for this project was provided by a UK Sustainability Challenge grant, and we're really thankful for that. Heather Wilson, John Saylor, and Nathan Skinner from LFUCG have been engaged throughout this process and really helpful in helping us access the sites and think about the kinds of data that we wanted to collect from these sites. And I mentioned earlier that they are doing ongoing work to manage these sites, and so we'll continue to partner with them to monitor these sites over time and do additional work that that will hopefully benefit them and the research. All of my interns are listed here: Kiernan, Sabrina, Kaylee, Julia, and William Witt. All did really great work over the summer, and they are forestry students, wildlife minors, and NRES, so really great sampling of students. John Cox, John Lotka, and Zach Hackworth all provided really critical help on the project, and so thankful for them. Soils were analyzed at UK Reg Services, and water was analyzed by Millie, and so we're really thankful for all their help.